Welcome to the Persephone training video covering maps, tracks, and basic operations. Part 1, Maps. Select the Maps tab. Select a map set from the upper navigation pane. A map set is the result of a single study of a single organism. A physical map is a representation of a sequence chromosome or a scaffold. A genetic map is a linkage group of markers not based on sequence. Right-click on a map set to display the properties, which will include the source of the data. To display a map, click on the map name. As you click on other maps, they will replace the existing one. To display more than one, pin them down by checking the box next to the map. You could move maps around and zoom in and zoom out. If you want to get them back into frame, click on Auto Arrange. To close a map, right-click on it and select Close Plate. To close all plates, hold down Clear All Plates on the main toolbar. To have maps pinned automatically upon selection, go to Tools, Settings, and check Auto Pin Down Newly Added Plates. Part 2, Tracks. Plates can contain one or more tracks, a base track on which the map is built, and additional tracks from supplemental data which has been uploaded. Hover over the top bar of the plate to see the names of the tracks. Moving the cursor over the name will highlight the corresponding track. Clicking on the name will hide or display the track. The indicator above the track has three states: lighted when displayed, off when hidden, and blinking when loading. To hide one track in particular, right-click directly on the track and select Hide this track. Right-clicking within the plate and selecting Show Hide Track will also hide and display tracks. Additional tracks to be displayed by default can be defined for a map set. Right-click on a map set and select Define Default Tracks. Part 3, Physical Maps. A physical map consists of nucleotides with base pair coordinates. To zoom into a specific region, hold the Shift key and select the region with the mouse, then release. Zoom into a specific point on the map by double-clicking on it. Or zoom in and out by rolling the mouse wheel. At the gene level, an arrow pattern indicates the orientation. Click on a gene to open the annotation details. The Basic Properties tab contains the method of gene prediction, IDs, and coordinates. There's a Protein Sequence tab, Splice Sequence, and the Unspliced sequence, which displays introns in blue, exons in red, and additional flanking nucleotides can be added in green. Qualifiers display gene details which can contain hyperlinks to external information about the gene. The orthologous relations in our case were defined using reciprocal BLAST. The calculation is done outside Persephone, so the ortholog data stored in the database can be prepared using other algorithms. Part 4, Genetic Maps. A genetic map with centimorgan coordinates is displayed in red with a track of markers and, if available, QTLs to the right. Click on a marker to open the marker details. The Basic Properties tab includes IDs, coordinates, and source information, and when available, qualifiers or marker aliases and sequences. The All Locations tab lists other locations where the marker can be found and their qualifiers. When the number of markers is too large to visualize all at once, you can create a new track based on a subset of markers. Right-click on the plate and select Create New Track. In the Genetic Map filter window, choose to filter by marker name, marker type, source organism, assay type, source, or qualifier. Previous filter settings are retained, so be careful to update them when creating new tracks. Data Export. Right-click on a plate and select Export Track. Select the columns to export from the Fields list on the right. You can filter by coordinates or by selecting rows in the tables. Choose to export just the selected items or export all. You will get a tab-separated values file. For faster results with larger datasets, pre-select a region of the map by holding the Control button while selecting with the mouse. Right-click on the region and select Export Track. You can see the coordinates to the subset that you've selected. Pre-selection is especially important with physical maps because of the number of records involved. Notice how long it takes just to add one field to these 9,000 records. By pre-selecting first, we can minimize the time it takes to export our data. To export a complete map set, right-click a set in the left navigation pane and select Export. This concludes our training video covering basic operations in Persephone.